In this video, we'll be taking a closer look at facilitated diffusion as it pertains to membrane transport and passive transport of materials. We'll also be looking at active transport of materials using facilitated diffusion. So again, facilitated diffusing is the use of a protein channel or a carrier protein in order to facilitate the diffusion of materials that can't normally pass through the cell membrane. From a high concentration to a low concentration, using like a protein channel here. You have just some, maybe large or small polar molecules. Water in large quantities is going to use a channel called an aquaporin. It's just a pore, which is a hole in water. Aquaporin. Charged ions, sodium, potassium, are examples of that constantly are going across some sorts of cells. They will use a protein channel in order to do that. Active transport is going to, when it's those materials are moving against the concentration gradient, and there are special proteins called protein pumps or pumps that are going to be used, and these require metabolic energy. These are going to require ATP. They have to be hooked up to something. You think about it that way, and so it's kind of like the ATP is going to provide power to power that pump to do the work. The work here is going to be to establish concentration gradients, moving things into a high concentration so that that concentration gradient can be later used to do some sort of cellular work. Some kinds of facilitated diffusion are called co-transport. And co-transport is, also called secondary active transport. And this is going to be where it uses energy from an electrochemical gradient to transport two different ions across the membrane through a protein. And so there is symport, where you have two different ions that are going to be transported in the same direction here. So maybe this S1 helps S2 get across. S2 can't get across unless it hooks up with S1. And so sometimes you'll see a situation like that. We'll see a couple of examples of that in another unit. And then this is antiport, where they're going opposite. That sim means together, anta means opposite. And so S1 goes through one way, S2 goes another way. We'll talk about something called a sodium-potassium pump, for instance, where sodium is pumped one way and potassium is pumped the other through the same mechanism, through that same proton pump or protein pump. The next one is we're going to look at is this idea of membrane potential. This is where the cell membrane allows for the formation of electrochemical gradients. And so when we're talking about electrochemical gradients, we're talking about electrical charge, right? So positive and negative charges building up across the membrane, and the membrane itself has a potential energy. And so this is often called membrane potential, where there's like a voltage potential across the cell. If anything trips that voltage potential, then it causes there to be a release of energy. Like in a nerve cell, this would be called like an action potential when those gradients switch in a certain way. Membranes can become polarized when those ions move. There's some sort of trigger that causes the ions to break those concentration gradients and move one way or the other. So here you have a picture of a concentration gradient, the high down here, low here. Something triggers that membrane, causes it to release that potential. These ions will go across and reach equilibrium, and then they need to be brought back to that potential again. Here's a picture of the one we mentioned earlier, the sodium-potassium pump. Sometimes you'll see it called sodium-potassium ATPase, and this contributes to the maintenance of that membrane potential. Once sodium and potassium switch places through just regular passive diffusion, releasing that energy, then they have to be put back in order. Think of like when you set a mousetrap, that mousetrap releases a ton of energy when the mouse steps on it. Well, and then you have to go back and reset it, right? And so the sodium and potassium pumps are resetting that mousetrap, so to speak. They're resetting the membrane potential, which allows it to work again. And you can see kind of what's happening here. 3 sodiums are going to be pumped across the gradient, and at the same time, the two potassiums are being pumped across the gradient as well in order to establish that membrane potential.